Hello everyone. Today I'll be explaining bacterial gene regulation. This is a key concept in microbiology that shows how bacteria control which genes are turned on or off depending on their environment. Gene regulation is basically the ability of a bacteria to turn genes on or off. This is important because it saves energy and allows bacteria to adapt quickly to changes, like when nutrients are available or when conditions change. Without regulation, bacteria would waste resources making proteins they don't even need. The main way bacteria regulate genes are through something called an operon. An operon is a cluster of genes controlled by a single switch. It has three main parts: the promoter, where RNA polymerase binds, the operator, which acts like the on and off switch, and structural genes, which code for the proteins. This setup allows bacteria to control multiple genes all at once. An example would be lac operon. The best example is the lac operon in E. coli. When lactose is not around, a repressor blocks the operator and the genes stay off. But if lactose is present, the repressor is removed and the genes switch on. The bacteria then makes enzymes to digest lactose. This shows how bacteria only make proteins when they are actually useful, which conserves energy. There are two main types of control. Inducible systems like the lac operon are usually off but can be turned on when needed. Repressible systems like the TRP operon are usually on but get turned off if the cell already has enough of the product. This balance makes sure bacteria don't waste energy making something they already have plenty of. Overall, gene regulation is critical because it helps bacteria survive. It lets them conserve energy, respond to changes, and adapt in tough conditions like low nutrients or stressful environments. This ability is one of the reasons bacteria are so successful at living in almost anywhere on Earth. Here are the sources I used for this presentation, including my textbook and some classic research on gene regulation.
Hello class, my name is Hope Durham and I will be walking you through the process of DNA replication. This is one of the most essential steps in cell division. Without this process, cells would not be able to pass on genetic information accurately to new cells. Replication begins with initiation, the first step. When specific sequences of DNA, called origins of replication, they signal where the process should start. At these sites, the DNA double helix unwinds and separates into two strands. The enzyme helicase is responsible for breaking the hydrogen bonds that hold the base pairs together, creating what we call a replication fork. Once the DNA is open, another enzyme, primase, adds short RNA primers. These primers act as starting points because DNA polymerase, the main enzyme that builds our new DNA, can only attach nucleotides to an existing strand. Then comes the elongation stage. DNA polymerase adds complementary nucleotides to the exposed bases, always working in the 5 to 3 direction. This creates two different situations. On the leading strand, DNA is sensitized continuously toward the replication fork. But on the lagging strand, synthesis occurs in short, discontinuous segments known as Okazaki fragments, which are later joined together. To finish, DNA ligase seals the gaps between fragments on the lagging strand, ensuring that both new DNA molecules are complete and continuous. Finally, in the termination phase, the last phase, replication ends once the entire molecule has been copied. The result is two identical double helixes, each with one original strand and one newly synthesized strand. This is called the semi-conservative model of replication. DNA replication is crucial because it ensures genetic stability every time a cell divides, whether in growth, repair, or reproduction. It guarantees that each daughter cell has the exact same instructions. Mistakes in this process can lead to mutations, which sometimes cause disease, but overall, this highly accurate system is the foundation of life itself.
Hi, my name is Brittany, and I'll be talking about the importance and applications of DNA replication. Why is DNA replication important? It is important in growth and repair of cells. DNA replication allows for cells to divide. This is very important to the survival of all living organisms. This process grows single fertilized eggs to form a mature body. It can also heal and replace damaged or old cells. It passes on genetic information. When cells are dividing, the new cells will need to be paired with an original strand, which has the genetic information for the new cell to follow. DNA replication makes sure that each new daughter cell gets a copy of the parent cell, so they can then function properly. Adaptation. Sometimes in the process of replication, there can be small errors called mutations. Mutations have helped humans evolve and adapt throughout the years. Applications of DNA. Medical and forensics. It can medically be used to diagnose diseases, genetic disorders, and detect infections. To diagnose diseases, it sometimes requires DNA extraction, and an example would be Down syndrome. In forensics, DNA evidence can be collected and be able to solve crimes. Vaccines. DNA can help in creating vaccines that control and stop certain diseases. DNA human vaccines are still in the development, but are often used in various animal vaccines.
All right, my name's Abigail Tilton, and I'm doing horizontal gene transfer, specifically transformation. So, horizontal gene transfer is how bacteria share DNA with each other outside of normal reproduction. One way this happens is called transformation, which is when a bacterial cell dies and picks up loose DNA from its environment. This process was first discovered in 1928 by Frederick Griffith when he noticed harmless bacteria became harmful after picking up DNA from dead bacteria. The process of transformation starts with a donor cell when it dies, and its DNA is released into the environment. A nearby recipient cell can take up those DNA fragments if the DNA is compatible. It can integrate into the recipient's chromosome. Once that happens, the recipient cell now has new traits. The importance. Transformation is important because it allows bacteria to adapt and survive in new environments. Unfortunately, this process also spreads antibiotic resistant genes, which is a major medical concern. On the positive side, scientists use transformation in research and biotechnology. For example, we use it to insert human genes into bacteria so they can produce medicines like insulin. And here are my references.
Hello everyone, my name is Jaden Portland, and my topic for this portion of the presentation is horizontal gene transfer, specifically focusing on transduction and conjugation. Horizontal gene transfer is how bacteria share genetic information without reproduction. Two of the most important mechanisms are transduction and conjugation. Starting with transduction, transduction happens when a bacteriophage, which is a virus that infects bacteria, accidentally packages bacterial DNA instead of its own viral DNA. When this phage infects another bacterium, it transfers that DNA into the new host. There are two types of this. First, generalized transduction, where any bacterial gene might be transferred, and second, specialized transduction, where only genes near the vital integration site are transferred. This process is especially important because it allows bacteria to acquire new traits like toxin production or antibiotic resistance. Moving on to conjugation. Conjugation is different because it requires direct contact between two bacterial cells. A donor cell with an F plasmid, called F+, uses a pilus to connect to a recipient cell, F-. This plasmid DNA is then transferred into the recipient, which may also gain new traits, including antibiotic resistance. Conjugation is one of the main reasons resistance spreads quickly through bacterial populations. Together, these processes contribute to genetic diversity in bacteria and play a major role in public health challenges like antibiotic resistance.
Everybody, my name is Nhi Nguyen, and today I'll cover plasmid and transposon. So, what are plasmid? Plasmid are extrachromosomal DNA that exist separately from the material chromosome inside a cell. They're circular, double-stranded, and supercoiled. They're important because they can carry up to several hundred of genes that can code for extra traits, such as antibiotic resistant or toxin production. They also replicate independently, so that's why they can spread the resistance so fast from one bacterial cell to another one. Now I'm going to move on to transposon. So what are they? Transposon could also be called jumping genes. They're DNA segments that move within the genome. There are two main classifications for transposon. The first one is retrotransposon. They use the copy and paste method through an RNA before they make the DNA copy. Our second class is DNA transposon. They use the cut and paste method through an enzyme called transposase, and some of the transposon, they're autonomous, which mean that they can move themselves, but some of them, they're non-autonomous, which mean that they would need help from a partner. In conclusion, plasmids are extracircular DNA that bacteria can share, often spread traits like antibiotic resistant. Transposon, they're jumping genes that can reshape the genomes through copy and paste or cut and paste method. Together, they show us how microbes can adapt so quickly, exchange DNA, and gain survival advantages. They're clinically important because they drive antibiotic resistance, which make our treatment much harder. And studying these mechanisms can help us gain a better understanding and design better strategies to control infection and protect our public health.
Hi everybody, my name is Piper. I'm a part of group A2, and today we're going to be talking about microbial metabolism and genetics. This topic is really important because it explains how microbes live, grow, and influence the world around us. First, I'll cover microbial metabolism, which is basically how microbes get energy and build the components they need to survive. Then I'll move into microbial genetics, which looks at how traits are stored, passed on, and sometimes changed. Together, metabolism and genetics gives us the full picture of how microbes function and adapt. This matters because microbes impact so many areas of life: our health, the food we eat, the environment, and medicine. For example, understanding microbial metabolism is key for developing antibiotics and biotechnology tools. It also helps us face global challenges like antibiotic resistance. Finally, I'll talk about mutant selection. Scientists study microbial mutants to understand what specific genes do and how metabolic pathways work. This research is really valuable because it helps us track resistance mechanisms and even develop new treatments for disease. So overall, microbial metabolism and genetics aren't just abstract concepts. They help us understand how microbes in a way that leads to real-world solutions in health, science, and the environment.
I would like to close our presentation with Christian worldview. I found this one verse from Genesis chapter 1, verse 27. So God created mankind in his own image. In the image of God, he created them. Male and female, he created them. It shows us that every person is created in God's image, giving them the inherent dignity and value. Research in healthcare practices should reflect love, care, and respect for all individuals. Ethical responsibility in microbiology should include ensuring safety, protecting life, and advancing knowledge in the common good. For application, in studies genetic microbes and diseases, scientists are called to apply their knowledge responsibly. Careers in microbiology, like medicine, research, public health, should prioritize human flourishing, especially caring for vulnerable population. Well, thank you so much for listening, and I hope that you will do good on your exam next week.